Today I have kind of an ambitious, aspirational talk about the value of natural history collections, this very broad topic. I will mostly be talking about rats with a brief sojourn into shrews and talking about new technologies like genomes and cellular biology that are helping revolutionize both historical collections back to the 1800s and contemporary collections. So natural history collections house a vast record of biodiversity on our planet. They house the type specimens upon which nearly 2 million species names are based, and 21st century advances in sciences are, from morphology to genome sequencing and cellular biology, as I said, are vastly increasing their value to help us understand and respond to our dynamic world. So the rich historical value of museums does not constrain the contemporary imperative to collect new specimens, which is embodied by many of the staff at this institution who are just avid collectors, and that's a really rare and special thing to have people who are going out and still building biodiversity collections because they're extremely important to us, understanding and responding to contribute to saving biodiversity. I was trained partly at the University of California, Berkeley, twice as an undergraduate and as a postdoctoral researcher, and I was trained with the aspiration that collections have value spanning centuries. So Joseph Grinnell, the first director of the Museum of Vertebrate Zoology at Berkeley, worked during the rapid expansion of industrial agriculture and human populations in California, and then he realized that the specimens he collected by museums would be valuable reference for centuries to come. The Grinnell Resurvey project, led by the Museum of Vertebrate Zoology, is one legacy of those efforts. It involved revisiting in the 21st century sites surveyed in the early 20th century. That project has led to numerous papers across mammals, reptiles, amphibians, and birds. This is just one of them that I was involved in as a postdoc there, and it involved records of 30,000 mammal specimens from nearly 300 sites across the across elevational transects of California's central mountain ranges. It revealed a century of climate change impacts on the distributions of species ranges, leading to upward expansions, but heterogeneous responses of species across their range, decoupling communities, and leading to major changes in species assemblages. The historical specimens from that project also revealed the genetic impacts of a century of climate change, loss of genetic diversity in the contracting range of the alpine chipmunk, the species you see here, which was one of the species that was retracting upslope the most in the southern parts of the Sierra Nevada mountain ranges, revealed increasing population structure in the species, increasing isolation in the species from the historical specimens to the contemporary specimens. So with the common call of a biodiversity crisis, the need for investment in biodiversity discovery, or as we know it, taxonomy, is greater than ever. This is a plan that was put together by the Australian and New Zealand equivalents of the Academy of Sciences. It's a 10-year plan highlighting the need for biodiversity discovery. And as this study, or this report, which has many more details than I can cover here, shows us, the vast majority of species on Earth lack a scientific description, with only about 25% of species, even in Australia, described, and that the need for new descriptions and new taxonomists is imperative. So they also commissioned, as part of this report, an economic report by Deloitte Economics to look into the economic value of taxonomy, and they found that for every dollar spent in taxonomy, it resulted in between $4 to $35. And I often get many questions about how they got to that number. I'm not the economist who did this. This is a much longer report than I can show you here. But these are the kind of things we need to communicate to funding bodies, governments, and other sources to advocate for the need for new taxonomy, because most of the scientists in the room can tell you how challenging it can be to fund taxonomy, the very basic science of understanding the diversity of life we have on our planet. Take, for example, mammals. This slide takes a moment to load because it's got quite a few dots on it. Every dot on this slide represents an individual mammalian species, all 6,508 species. At least that was the case in February 2020 when I made this slide, but 4 years later, that number has increased by 245 species. There's probably been an increase while I've been on this trip. So you can see that mammal taxonomy is growing greatly. In 2005, which was the last edition of the Mammal Species of the World, that we recognized more than 1,300 fewer species of mammals on Earth. So we are still describing the limits of living species on our planet. As noted by Grinnell, the value of natural history collections is wherever we work. And because biodiversity is everywhere and increasingly in remote and isolated areas away from human occupation, where we often work in collections is where others do not. This map shows where Australian mammal collections have worked. They comprise more than 300,000 specimens, not an enormous number by global standards, but an important number with many important taxa and species. They're housed in 9 museums and sampled from across the breadth of the continent and mostly from nearby surrounding regions. For the past 14 years, I've worked with the Museum Zoologicum Bogoriense, formerly in the Institute of Sciences, which is now the Indonesian National Innovation and Research and Innovation Agency, BRIN. And our goal is to survey remote areas of Sulawesi and the island of Sangir Besar, which is halfway really between Sulawesi and the southern part of the Philippines. Our elevation span between 200 to 2,800 meters, with mountains on Sulawesi getting slightly over 3,000 meters, but so we're approaching the highest elevations on the island. I came to collect the rats, and thanks to Jake Esselstyn, the shrews. The rats contain several genera for which we had no genetic samples, and we really uncertain how they fit into phylogenetic trees, and we thought they might hold clues to the evolution of rodents in Australia, given the location of Sulawesi. The shrews were poorly sampled, and with few specimens to define the limits of species. When Wallace visited Sulawesi in 1856, he reported 14 species of terrestrial mammals and 7 bats. Among these, he noted Sulawesi's endemic babirusa, anoa, macaque, tarsier, bear cuscus, and squirrels. But he did not mention any of the rats, including several specimens that he collected of rats and which are now type specimens of those species, which I only noticed at the NHM a few weeks ago. So today we know there are at least 80 species of rats and shrews endemic to Sulawesi, so found nowhere else. Since 2016, we have worked with a multi-taxonomic team led by Bryn and the University of California, Berkeley, to survey vertebrates, invertebrates, and their parasites, very similar to much work that happens here in Africa and elsewhere. So it looks something like this. So this is where we work, this is where we have to do science, we have to hike in, and you can also see that I've aged a little bit in the last 10 years. For much of this crowd, I may not have to describe shrews. I often have to describe them when I'm in Australia, since they're not present in Australia, but they're common members of ecosystems on all continents except Antarctica and Australia. The genus Crocidura is the most species-rich genus of mammals on Earth, and they reach as far as Sulawesi, but do not get to Australia, where they're replaced by a marsupial-like, shrew-like marsupials in the family Dasyuridae, small carnivorous marsupials like Antechinus and Dunarts and Planigales. When we started on Sulawesi in 2010, there were 6 species described from the island. Through nearly a decade of collecting and by very detailed work mostly by Jake Esselstyn, and morphological and genetic comparisons, we have increased the known number of species to 21, so more than 3 times as many species as were thought to be present of mammals. Small, cryptic mammals, but still fascinating. And really it took new collections, new specimens. We had maybe less than 100 specimens to work with when we started, and we've added well over thousands of specimens. Our collections also increased the number of rats known from Sulawesi with 17 new species and 6 new genera. And those genera include sinking 2 other genera. So anyone who studies Southeast Asian rodents, the genus Maximes, which is widespread from Thailand and Malay Peninsula through Indonesia into the Philippines, has been sunk now into chronomes in about a week or so. Sorry for that. Most of these were not known in any collections, and some were quite extraordinary, including this worm-eating rat, Posidentomes vermidax, known to the Mamasan people as sapiu. It is unique among nearly 2,700 species of rodents and having absolutely no molars, no chewing teeth, and having incisors that are used to snip up earthworms rather than being able to gnaw and chisel away. So departing from the key innovative toolkit of rodents, these ever-wearing chisel-like incisors that allow them to access food. So something on Sulawesi made it depart from being a rodent. They include charismatic animals like this, saltatorial hopping, also arthropod feeding hog-nosed rat, and remarkably an amphibious rat, the only amphibious mammal, completely unknown to science, known to the Mamasan people as balawai, or water rat, and kept in their homes as a talisman against fire. And the one and only specimen that we have in any collections is this one here, brought to us by Herman here, who after living with us for 3 weeks thought, this is really valuable, but maybe these scientists don't know have ever seen this thing, and presented us with the first amphibious mammal from Sulawesi. So the diversity of life is the heart of collections and taxonomy. We understand it as a phylogeny, a tree that reveals the history of species and their relationships. The systematic relationships are fundamental to many biological inferences, including geography, ecology, trait evolution, and speciation. So placing species in a phylogeny guides our taxonomic inference and reveals the richness of life. While mammals are not a particularly diverse clade in the tree of life, the subfamily Murinae and the family Muridae as well are exceptionally species-rich, comprising the subfamily Murinae comprising 11%. 1 in 8 mammals on Earth is a old world rat or mouse. Their diversity is recapitulated throughout their evolutionary history, so especially compared to their sister clade, which you can see in red in each of these stages. So the Rodentia make up about 40% of mammals. They're exceptionally diverse compared to other mammal orders, but particularly compared to the rabbits and pikas, their sisters, the Lagomorpha. Same with the superfamily Muroidea and its sisters, the hopping mice and jerboas and Dipotidae. And throughout the history, Murinae has always been the most diverse. Arguably within Muroidea, the Chrysetidae of South America and America are almost as diverse. Their sister group, the Nesomyidae from Madagascar, are much less diverse. The diversity of Murinae is distributed across most of the Eastern Hemisphere, with a particular concentration in the geologically complex Indo-Australian region, focused here on the right, where 2 continents and numerous oceanic archipelagos have been in a state of collision for millions of years. As Australia has drifted away from Antarctica, collided in here, forming New Guinea, forming Sulawesi in between them, and crashing into the, slowly crashing into the Asian continental shelf, with the oceanic Philippines emerging to the north through its own processes. This phylogeny of the Murinae is organized in tribes, and colors reflect geographic distributions. You can see that much of the geography is obviously nested within this phylogeny, so it's relatively interpretable. It is sufficiently complete such that we can place all but 3 of the genera in a phylogenetic context. And if you'll bear with me for 2 minutes, I will show you now all 142 genera. So I hope that leaves you with a sense of the biodiversity within Murinae, albeit only the genera, right? And with all the popularity of Pokemon, I mean, it has nothing on the natural world. And of course, mammals are far less, a small percentage of all the biodiversity on Earth. But we have the capacity now to start to understand. We know 2 million species on this planet. We estimate there's another 8 million more to describe. They deserve their recognition. Remarkably, 7 of the genera in that slide have only been recognized since 2021, most of them by members of this, described in partnership with members of this audience. And thank you to those team members who provided photos that allowed the genera to be shown. But it just shows that going out and collecting new specimens, and particularly these new genera have come not just through old collections, but new collections as well, and including new species descriptions. So it underscores the value of going out collecting new specimens, interrogating old specimens in new light. So again, where we work is often driven by our aspiration to realize the geographic origins of species and how it shapes their evolution. Through our collections from Sulawesi and with our phylogenetic understanding of murines, we understand that their diversity on Sulawesi was assembled by at least 7 colonizations across Wallace's line from the Asian continental shelf to Sulawesi, an island that has never been connected to continental Asia in its entire history. These spawned numerous colonizations of other oceanic archipelagos and continental landmasses, including at least 2 back colonizations through the Asian continental shelf, giving rise to diversity in the Asian continent, and 2 colonizations of New Guinea, leading to continental radiations in Australia. So our initial sense of going there was going to help us understand the radiation of Australian rodents was correct. This geographic understanding is the foundation for understanding repeated radiations of murines, or in any group, these repeated radiations are an opportunity to study the evolution of groups, especially where there are convergent ecomorphological examples. So remember, for instance, this worm-devouring rat, Posidentomys vermidex, from Sulawesi, remarkable among rodents and having no molars and incisors that cannot gnaw. Well, there's another genus on the Philippines, Rhynchomys, also a worm-eating rat. It has a couple of molars, so Posidentomys just beat it there. But they also have these incisors designed for stabbing, piercing, and cutting apart earthworms. The 2 are remarkably convergent in the elongation of their skulls, however, they're not particularly closely related. They're not more closely related to each other than either is to the lab mouse or the lab rat. So the same genomic variation that separates the lab mouse from the lab rat is at the root of the divergence between these 2 earthworm-eating rats, and with an ancestor somewhere near the base of Murinae 14 million years ago. So they underscore the strength of selection and unique geological circumstances in the Indo-Australian archipelago, in the Philippines, and in Sulawesi to reinvent rodents again and again. There are numerous other convergent examples. Here's that amphibious rodent, the water rat, Balawai, Wyomys, from Sulawesi, known only from the single specimen. It's highly convergent with montane water rats from New Guinea, with completely independent origins. They both have reduced ear sizes, soft fur, small eyes, and these extensive vibrissae on the underside of their tail that presumably is involved in swimming and in the water. But they're both independently evolved from terrestrial animalivorous or carnivorous rodents from Sulawesi, including Posidentomys and Melasmathrix, shown here from Sulawesi, and Pseudohydromes from New Guinea. The Philippines also has terrestrial animalivorous rodents, like Rhynchomys and Soricomys, shown here, but as yet no amphibious rat has been discovered, but who knows. So they also harbor small arboreal mice, including Heromes from Sulawesi, Lawrencomys from New Guinea, and Musaromys from the Philippines. And each of these slides you can see the phylogeny is depicted here and shows the origins of each of those clades independent. They include arboreal giant white-tailed rats, Bulimus in the Philippines, Euromys and Pteromes, which one of the species used to be in the genus Pyromys, which we sank and unfortunately broke up that obvious nomenclatural connection to Euromes, but these are very similar rats. They include woolly, folivorous, arboreal rats like Malamies in New Guinea, Aeropeplos, and Lenomes in Sulawesi, and the largest murines of all, Phloiomes, which get 2 kilograms from the Philippines. So each of these ecomorphological forms evolved with radiations in their respective land masses. So consider the 2 largest radiations on Sulawesi, the Echyothrix division, including Posidentomes shown here, and the Bunomes division, including Aeropeplos, as shown here. So while all but one species in the Echyothrix division are animalivorous, they have striking disparate ecological and morphological variation, such that in 2005, prior to any phylogenetic analyses in this taxonomic revision of mammal species of the world, they were split into 3 separate divisions and not considered closely related. But phylogenetic analyses revealed that they're from single radiation. Also remarkably, at least half the diversity in this clade was unknown until 21st century descriptions and new collections. The Bunomes radiation was not the first arrival to Sulawesi by rats, but it is the most diverse and includes a wide range of diets, body sizes, and locomotor adaptations for climbing or living on the ground. It includes that arboreal Teiromes, or formerly Pariromes, that's more similar to Euromes and Bulimus, and it includes these giant woolly rats, Aeropeplos and Lenomes, that are similar to Malamies and Phloiomes. A brief sojourn to linking genomes and phenomes, which may come a little bit early compared to some of the genome stuff, but part of what we want to link in these radiations is their phenotypes and their morphology to their evolution. I'll give you a brief example with our attempts with Posidentomes and Rhynchomes. So as I said, they're very obviously convergent earthworm-eating animals. That's evident in their skulls. So this is a plot from unpublished from my collaborator Pierre-Henri Fabre from the University of Montpellier, and it's 2D morphometric data from skulls of most murine genera, and you can see the cloud is mostly on the left, and that both Posidentomes and Rhynchomes, their skulls are occurring in a space unoccupied by any other murines, complete novelty, and converging in that new novel morphospace. We had access to over 14,000 protein coding genes from these 2 species, and thought this is a great place to test whether there's any correlation in their molecular evolution. Is there a rate shift associated with becoming an elongated snout? And while across murinae we found the usual pervasive positive selection in genes associated with immunity, reproduction, diet, we did find more genes with positive selection in lineages shifting to derived diets like carnivory or herbivory. Most murines are omnivorous. The base of murines is omnivory, but there have been lineages that have become fully carnivorous or fully herbivorous, and those showed higher numbers of genes under positive selection. But we did not find any genes with more shifts, rate shifts in their evolution, convergent between these 2 species than by random. So a little less satisfying. And I'll give you a more recent example. This is a paper that just came out last week, and it uses a method called Ornstein-Uhlenbeck modeling, and to identify parts of the tree where you have clades that are moving to a new selected optimum. So there's a new state that they're trying to achieve, and here we were measuring relative testes mass. So the relative testes mass is a proxy for sexual competition, and so when you have larger testes, when you're under more sexual competition, you expect to have larger testes, produce more sperm, and that's been proven in many other forms. And here we saw repeatedly across the tree shifts to selection for a smaller relative testes mass. And then when we looked at these same proteins in those species, what we saw was relaxed selection on genes that were under pervasive positive selection in the other species under sexual competition. And most of those genes, and all those genes were associated with spermatogenesis and sperm morphology. So this method derived from morphology of identifying where you have optimal shifts or shifts in selective forces might help us to pull out, rather than just looking at 2 convergent species and looking for genes. I think this is going to help us identify more areas of molecular evolution of interest. So yeah, these studies might be a little unsatisfying, but they're really only the beginning, and reiterate that collections give us access to genomes of millions of specimens, and the kind of large series needed for taxonomy to describe species are now give us windows into many detailed phenotypes from 3D models of their anatomy to try to dissect how phenomes and genomes evolve together. And then before I leave wherever we now work, partly because this is relevant to anyone who's collecting nowadays, and particularly the collectors in this room, is that museum specimens are contributing to underdoing wildlife disease surveillance. So particularly because we work in some of the most remote and often tropical regions where emerging diseases are most prevalent, our specimens have real value to uncover that. So as you know, we live in this tumultuous post-COVID world, and even before COVID, there were numerous papers written on the value of natural history collections and their role to play in surveying diseases and potentially emerging infectious diseases. On Sulawesi, our first disease surveillance focused on blood parasites, protists and trypanosomes and their relatives, and we screened using PCR methods over 2,300 rodents and shrews from 11 mountains. A quarter of those specimens were infected, and mainly by 2 species of trypanosomes. One, a native species or species complex distributed from Malaysia to Australia, and the second, an introduced species associated with the arrival of black rats, Trypanosoma lewisi. And the important finding of this study was that the prevalence of both species, whether introduced or native, was highest in the intact remote forests away from human occupation. And so that where we're going to work, where it may be where diseases are having the most novel interactions, so even introduced species are penetrating the most remote forests on Sulawesi, even though introduced rodents aren't really present there. In contrast, most wildlife disease surveillance is happening at the interface of human-wildlife interactions, such as villages and farms. And so we have a unique record in natural history collections as biodiversity scientists to add to that, complement that survey, and should be collecting samples that preserve viruses, bacteria, and other diseases. So Sanger sequencing helped us approach this nearly complete understanding of generic relationships in Murinae, but genomics, we need genomics to go further. So many projects are emerging globally to reveal the genomic variation of Earth's vast biodiversity. Bioplatforms Australia, which is the premier sequencing facility or umbrella for all of Australia, has had several initiatives aimed at providing genomic resources for Australia's wildlife. I've been a part of 2, the Oz Mammals Genomics Initiative and the Threatened Species Initiative. So the ambition of the OMG was to complete phylogenomic resolution for all recently living species of Australian marsupials, rodents, and bats. We've mostly done the rodents. The marsupials are coming. The bats are coming as well. And importantly, many of these species are rare or even extinct, and about 1/3 of species are only known from traditional museum specimens, such as study skins. So as I mentioned, the rodents of continental Australia, New Guinea, Australia, are the result of 2 colonizations from probably Sulawesi, but certainly from Asia. And they have radiated into over 180 species since their origin. The first colonists are in the tribe Hydromaini, and diversified into about 150 species across 40 genera, with many more species coming to be described. And they have a wide range of ecomorphological variation, from the most efficient kidneys of any mammal, this Notomys alexis hopping mouse in the bottom there, to amphibious rats, to giant woolly rats, to graminivorous, grass-feeding, vole-like rats. The second invaders are the Rattus, which arrived in New Guinea about 1 million years ago and quickly spread to Australia, radiating into approximately 30 species, but with more species to be described. Our phylogenomic work has relied on target enrichment focused on capturing exons either a custom set of exons designed to be orthologous across mammals, particularly marsupials, bats, and rodents, and a exome set, which is more like a half exome set that captures around 14,000 genes, or at least we get 14,000 usable orthologous genes out of this data set. OMG helped us do about 90% of the Australopapuan species. That's been published 2 years ago with a bit more data on ratus to be published, and with support from the NSF grant and partners at the University of Montana and Louisiana State University, we've done whole exome sequencing across 80% of genera in urinate. So this phylogeny, the Australopapuan rats, a version of which was published in Current Biology, reveals the contribution of historical specimens shown in red to resolving this tree. Many of these are skins from rare species that are likely still alive in the wild, but others are extinct. There are only remains preserved in museum collections or fossil deposits. So this phylogeny published in 2021, the year before the Current Biology paper, included 50 species of Australian rodents and 8 extinct species. So their extinctions amount to loss of about 10 million years of evolution within the past century and a half. And for many, their collapse was rapid and they were barely documented before they disappeared. One of them is this small mouse here, Pseudomys gouldi, Gould's mouse. Was first collected by the HMS Beagle, not by Darwin, but by other naturalists aboard the ship in 1837, and then last collected by our museum's first curator of zoology, Wilhelm Blandowski, in 1857 from the northwest desert corner of Victoria. When the species was then presumed extinct, but when we sequenced it, it showed a very close similarity to a living species from the far coast of Western Australia, with limited genetic divergence between them, well within the variation seen within other species. And this minimal divergence was particularly striking because the other species lived across the continent and survived on a single island off the coast of Western Australia. So while we had removed an extinct species from these, so taxonomic precedence meant that Pseudomys gouldi was the name applied to both of these species, and they became Pseudomys gouldi. And while we removed an extinct species from the extinction list, it also demonstrated a continental collapse of a species. This is a mouse that was living in abundance across the southern half of Australia and then collapsed to a single island. And that collapse is evident in the genetic diversity measured by heterozygosity here in the living specimens of that species. So in 1857, when Blandowski collected the last mouse known in Victoria, the genetic diversity, heterozygosity, was comparable to other widespread species. And actually most of the extinct species, or all the extinct species, had genetic diversity that would be expected from a wide-ranging, continentally distributed species. Most of them collapsing to extinction, Gouldi collapsing to a single island with the lowest heterozygosity of any rodent in this study. So this phylogenetic sampling and collapse in heterozygosity over the last century, or the last 167 years, is a realization of the centennial aspirations of collections. And it asks us what other things we can ask of collections. So access to the centennial record of museum collections is increasingly being enabled by the availability of reference genomes, which we can now make for species across much of the tree of life. So through the Oz Mammal Genome Project, we produced a handful of scaffolded reference genomes, including the most useful one that we've done so far, which is this broad-toothed rat. Broad-toothed rats are like equivalent to European voles. They're related to other Australian rodents, part of the first invaders. They feed exclusively on grasses. They live in very wet meadow environments, which are a patchy environment in a matrix of forests throughout most of this system. Review of historical records and revisiting of sites showed that they had disappeared from more than half of sites in Victoria, and that their persistence was correlated with cooler, wetter environments at higher elevation, consistent with an impact of global warming. Those high elevation sites were hit very hard by Black Summer fires, including this site in Alpine, Victoria, which Ones contained hundreds of broadtooth rats. You can see their trails through the middle here, and these are trails that are passed down through generations. These animals used to run through what would have been very thick shrubby vegetation, but then, since scorched. In the 6 years of monitoring this site, the animals have yet to return. So given the catastrophe of the bushfires, the documented declines in potential abundance, we wanted to apply genome resequencing and genetic data to the species, and another endangered species that has a similar distribution but is associated with forest environments throughout the Southeast Australia. This is the smoky mouse, Pseudomys scumius. So for broadtooth rats, we reproduce whole genome resequencing data. So by having a reference genome, you can produce low quality shotgun sequencing, and you can map it to this reference genome and get really useful and reliable data from that species. We're able to do, and short read sequencing is what's particularly applicable to historical specimens. We're able to sequence back to specimens from 1918 up to the present and examine their change in structure and genetic variation. So our sampling is across most of the species range, which is largely restricted to Southeast Australia. The mainland population, that is excluding Tasmania, is federally listed as endangered and was the subject of the main study. The Tasmanian subspecies is listed as least concern, but has very little sampling. We could only cobble together 3 samples in any collections that were used in the study. When we analyze the data, the population structure, so up here you can see a PCA of the genetic variation that usually reflects your geographic structure, but then you can also use models. This is a structure analysis that will tell you how many populations in here you have, and actually the most likely number of populations is 1, so that these were really a highly connected population, even if there is some substructure that's apparent, and up to 4 populations makes geographic sense. So there's some maybe isolation by distance, but they're geographically sensible, even if the data tells us that this was a highly connected population throughout time. If we look at even at those 4 populations, the genetic diversity, the heterozygosity, is relatively stable across space, excluding, with the exception of Tasmania, which has a lower heterozygosity than any individual. Every individual from Tasmania has lower heterozygosity than any individual from the mainland. And then also, we see little change or no significant change in heterozygosity from time, from the historical samples, whether from 1918 or from the 2000s. Whether or not Tasmania, this lower genetic diversity, is something that's leading to a phenotypic consequence is yet to be determined. It may be related to their original colonization of Tasmania. Their low heterozygosity is both historical and contemporary, suggesting they may have lived with that for a long time. But this is something that we want to analyze with other analyses and more genetic data, more genomic data. The smoky mouse has a similar distribution to broadtooth rats, with the addition of this extraliminal population in western Victoria in the Grampians National Park. And similar to the broadtooth rat, where they overlap, if the population model suggested there was 1 population, but a strong break between those populations in the east to the Grampians National Park. But as with the broadtooth rat, there was sensible geographic structure up to 5 populations, including splitting the 2 mountain ranges of the Grampians, the Victoria Range in the west and the Mount William Range in the east, which are separated by only 25 kilometers and are as different from each other as any sample from the rest of the species ranges from one another. So for smoky mice, heterozygosity varied greatly across the range, maybe similar to the Tasmania. For broadtooth rats, where in the Grampians, this long isolated population had lower heterozygosity than the rest of their range. But we also saw a significant decline in heterozygosity from the 1970s, which were the oldest specimens we had in the Grampians, to the present. So much so that there's now some investment by the state in considering whether they need to do translocations to recover genetic diversity, particularly in the Victoria Range. So our centennial aspirations extend not just to the past, but to the future, what people are going to be asking of our collections. And that's why we continue, and the imperative to collect today is still important. Joseph Grinnell in 1915 published in the journal Science, the leading journal of our field, on his concern over the decline in collecting. However, it wasn't really till the end of the 20th century that there was evidence of the precipitous decline in collecting of specimens. And this is a plot based on collections in U.S. Natural history collections, but the pattern is reflected globally. Numerous papers have been published on the value and minimal impacts of collecting. This is one we published in our museum journal on the impacts of collecting vertebrates over 160 years in Victoria, and relative to the impacts of the other activities of humans, including road deaths in Victoria and duck hunters, which killed each year nearly 15 times as many birds as we've collected over the entire history of Victoria. And the sampling map here shows you how incomplete sampling is, even with all those specimens. So if anything, somebody should be mad at us for not collecting enough, not for continuing to collect. However, despite these efforts in places like Victoria, we are unlikely to collect any new whole birds or mammals unless they come to us as salvaged dead already. So we have to think about new ways of collecting. We need to think about this rich history of collection. I've shown you many examples of specimens, historical specimens, and how we've been able to get genetic data out of them, a little bit on how we get morphological data out of them, but there's much more we can get out of them. So for more than a century, whole mammalian specimens were collected primarily as skins, skulls, and other skeletal material, and wet, fluid-preserved bodies. Globally, these amount to about 10 million specimens, of which 6 million you could look up online on GBIF. And from the late 19th through early 20th century, these provide a rich record of mammalian diversity and distributions during the emergence of globalized human impacts. After nearly a century of collecting skins...
This is all mammals in the world?
Yes, yeah, all mammals in the world, yeah. Well, what's available on GBIF, and then a rough estimation of what's not on GBIF to get you to roughly 10 million. Not in Australia, we only have 300,000 specimens. So after nearly a century of collecting these preserved specimens, collections started to shift to collecting DNA-viable tissue samples, which were referred to as material samples, and they have increased to nearly 250,000 specimens globally as of September 2022. So one solution to documenting biodiversity is the rise of digital observations or machine observations through either camera work, acoustics, even citizen science. Those are the fastest growing record of mammalian species on GBIF or in almost any collection. It's something that we've adopted in our collecting practices following the catastrophic bushfires, investment in bushfire management and ecosystem resilience, particularly around prescribed burning. In the last 3 years, we've surveyed over 1,500 sites across Victoria, basically from border to border, assessing the impacts of fire, time-sense fire, and fire intensity on mammalian, avian, and vegetation communities. These resulted in more than 6 million images, which we've then registered more than 200,000 mammalian records. And for context, that's about 5 times the number of mammalian records that we collected in museums of Victoria over the first 170 years, so in the last 3 years. Of course, these are just images, and they lack a lot of the value of whole specimens. But at least we can provide a tangible record, and we can register those specimens and make them available online. So Australia feeds its data through the Atlas of Living Australia, where we can attach image records, and also eventually audio records. And then those feed into GBIF, where you can see the actual, just the species name records. The audio data we've collected, we've had to do manual listening to get out the 100,000 bird records that are here, but we now have funding where we're trying to leverage all the data. So each recorder produces about 4,000 minutes and from each deployment, and we've listened to about 21 minutes, which we found is equivalent to a person going and doing a bird point transect on site. So now we've got funding to try to leverage that data to pull out all the bird records and make sure that they're registered in databases. Because data, if it's not published or not registered, is lost. So you need to get it into collections or into publications. So today, many of the mammals we would have collected in Australia, in parts of Europe, in the US as well, as we would have collected skin, skulls, or fluid bodies, we are sampled only as material samples, most commonly for mammals, an ear biopsy, a small ear punch from their ear, and that provides something of a genetic sample, but it's very limited in quantity and quality. And of course, those ears are also made up of large amounts of fibroblast skin cells. And I'll talk to you about trying to collect cells from those samples. So the genomic studies of collections spanning the past 3 centuries have shown us the dramatic collapse in genetic diversity and some species genetic diversity that we wish we had still available to recover populations at risk. So that's part of the value and part of the inspiration for me of trying to collect cells. One was that we weren't going to be able to collect whole animals anymore, so I needed something more than just a ear snip and ethanol that would extract once. And 2, that we were losing genetic diversity for which all we had was dead DNA in the skin to recover. So one of the lessons was that cells improve genomes. They're a renewable source of DNA. And also, as we start doing genomes not all the same. You've heard me say whole genome resequencing, scaffolded genomes. A genome is not a genome, it's not a genome. Genomes come on a scale from just shotgun sequencing random fragments to building them into contigs to scaffolding to getting to what we call T to T, which is telomere to telomere, complete genome reference. The human genome only really, even though it was announced in 2000, has only really got to a T to T reference in the last few years due to new methods in gap filling and so forth. And those higher quality reference genomes, and the higher the quality reference genome, the better your data will be downstream when you do shotgun sequencing. Those are enabled by high quality DNA, as also having cellular resource. Being able to pull the nucleus out of a cell helps in some methods for building scaffolded references. Genomes improve and also demand cells. So part of this is in the concept of recovering lost diversity. So many zoos around the world, led by the San Diego Zoo, have been freezing cells of their collections since the 1970s, really. And even the San Diego Zoo has taken cells from a tissue of a black-footed ferret frozen in the 1980s, a female that never reproduced, and they've been able to, in the last 5 years, grow 3 new baby black-footed ferrets from those cells, the genome of that cell, by implanting them into a pregnant female. The other is these efforts of de-extinction, what I call engineering genomes from historical specimens. In our lab, we work closely with the Tiger Lab at the University of Melbourne that is attempting to resurrect the extinct thylacine by taking genomes from our specimens, sequencing them to great detail, and then editing the cell of another species, a fat-tailed dunnart, to make it appear like a thylacine. And it would have been much easier to start with the cell of a thylacine. And that's something that we can leave for future generations as cells of living species. It also allows lots of really cool biology of wildlife. I won't get into them too much, but things like mitonuclear interactions, the variation in the mitochondria and how it interacts with the nucleus affects species boundaries and the evolution of species and their ability to respond to climate change, because the mitochondria that's good for this environment and may not interact with the nucleus that's good for this environment, and when populations come together, that may decouple their response. It allows lots of wildlife disease research, viral transfection, all this kind of work you can do with cells. And a lot of wildlife disease surveillance was doing that during COVID. They were collecting cells of some animals, checking in their transfection, and throwing them away. We can have a resource for that. And then the other is cellular conservation biology. People are using methods to, like gene drive, to manipulate and put either beneficial sequences out in the environment. So the group we work with is working on cane toad toxic resistance in northern quolls. So this introduced cane toad you may know about has come in, and it carries very toxicity, and it's killing native wildlife in huge numbers, particularly northern quolls, which don't seem to like the birds. The birds seem to learn not to eat them, but the quolls don't, and they eat them and die. And they've been able to put in a cane toad toxic resistance into the cells of quolls, and at least in solution, those cells do not die when exposed to this disease. So they want to get that out into the environment. Might also help us with controlling pest species. So from a museum's perspective, there are 3 real major stages to collecting cells. One is how you do in the field sampling, then how you culture the cells, and then how you store them long term. The first and the third are absolutely essential. You need to have liquid nitrogen to store your cells long term, and you need to go in the field and collect them. And going in the field is wherever we now work. So we need methods that work where we go, where biodiversity lives. And I'll give you a short example with the smoky mouse. The species I say that lives isolated in Grampians National Park is one of its most isolated populations. There are two populations. We've been tracking the species for 12 years now, and it occurs only in these narrow ranges of the Victoria Range, about 6 kilometers there, and the Mount William Range, 25 kilometers apart. These populations are slightly structured. Both of them have seen declines in heterozygosity since the 1970s to today. To get to the species, we have to go out into the forest. There's no close town, there's no power. We have to camp to be close to our traps. We have to hike up to an hour into the site to find where the animals are. So we need methods that work for species in remote conditions like this. The first method is a method where we don't freeze the samples. This is where they're kept cool on ice in a culture medium, and that requires getting them back to a lab within 1 to 2 weeks to either culture them or slow freeze them, which I'll show you in a minute. And we've tested this method in smoky mice, and it works well in reptiles and birds. The other surprise as to how well this works on already dead animals. So as the state collection in Victoria, we receive dead animals from the Department of Environment and from the public, including beached dolphins, sea turtles, and this gannet. And these specimens, we don't really know how long they've been dead. We think 1 to 7 days, still can produce viable cells in the lab. The other method's important, especially if you don't have a cell culture facility, is to be able to freeze your samples down. All you really need for this is a -80 and ultimately liquid nitrogen. And that's where you get the primary tissue in. It's been kept cool and on culture medium. But then you can use these very simple foam devices to slow cool your sample down at 1 degree per Celsius. That stops the cells from exploding, which if you throw them in liquid nitrogen, any cell will explode, and keeps them alive, and then you can culture them at a much later date. And similar to this is a method of flash freezing them immediately into liquid nitrogen on the site of the field. And this is a method that is important when you don't have access to a cell culture facility in the short term. If you're going to be in the field for more than 2 weeks, as we often are in Indonesia, then you need a method that works in those remote conditions and keep the animals. You have to keep the cells from exploding, so you have to use a special cryoprotectant, which has a balance between being poisonous to the cells, but then perfusing enough into the cells to actually keep them healthy. But this is proven successful. With last year was the first time. So last year, July to August 2023, we surveyed Klabat Mountain at the eastern tip of the northern peninsula of Sulawesi, and Sangihe or Sangir Besar Island, halfway between Sulawesi and the Philippines, collected samples into liquid nitrogen. That liquid nitrogen dewar actually sat in Indonesia for 6 months. It wasn't exported until March of 2024 this year, and we are still growing cells from those samples that have been sitting in liquid nitrogen a year later. And what we hope to do is test how many years we can go, if we can go multiple years, because that means that anyone taking liquid nitrogen in the field now, even if they don't have a cell culture facility, can start putting away primary cultures, much like the San Diego Zoo did with that black-footed ferret female. They put away in 1989, not knowing what they would do with it, and now are able to make new pups. So I hope I leave you with the understanding that museum collections are both historical in their scope and contemporary in their practices. The specimens we study span centuries of collecting, and the materials we leave for centuries to come are ever more rich and valuable for future students of the future. In this century, that includes whole organisms, their genome sequences, remotely tracked individuals, and cryopreservation of living cells, and other resources unfathomable a century ago. These we leave from a growing list of the millions of species with whom we share this biodiverse planet. This work involves many, many, many people and collaborators. These are just a short list of some of the most important contributors and funding sources. There's far too many people to properly acknowledge, but it bridges many different fields and interests, from conservation managers to molecular biologists to cell biologists to universities and collections. And hopefully that's the kind of diversity we'll bring to bear on studying biodiversity. So, thank you for listening.
Kevin, for your talk. I think there are many people using museum collections, so maybe there will be many questions. Okay, so let's start from the left.
Thank you for a very nice presentation. It was very, very informative. I would just like to ask about the heterozygosity in Tasmania that you mentioned. So was it observed at some other species also, apart from rodents? And also, how do you plan to investigate it? Because you mentioned that you will probably do that.
Yeah, so we have a grant to try to do it in about 60 other vertebrate species in Southeast Australia, but it was well reviewed, but the response was it wasn't feasible. We don't really have that data for other species from this region. I saw an opportunity with bushfire funding, and there were species, the state had a genetic risk assessment, and they had identified species for which we didn't have genetic data. So both the broadtooth rat and the smoky mouse, I might have mentioned, had no prior population genetic data. And so they said, we need population genetic data. And I said, well, why not just do whole genomes? We have this reference genome, let's do it. So yeah, we don't, the short answer is we don't know yet. We have a couple of other rodents that we've done with other types of analyses in the area, but we haven't done the within species analyses like this that show us how the heterozygosity is shifting. So the other analysis we want to do is where that heterozygosity is something that has evolved recently or over a long period of time, and there are these methods for looking at populations through time using Markov chain like the PSMC and MSMC methods. But they really rely on having a very good estimate of runs of homozygosity. So that means every SNP is called, and you know it's not heterozygous. And if you're dealing with genomic data, you need really high coverage to that. So these were basically 10x coverage. So we have, for every base, we have on average 10 reads. And so you can imagine when you're trying to call a heterozygote, if you're calling, if there's 5 of one base and 5 of the other, clearly a heterozygote. What if there's 6 and 4? What if there's 7 and 3? What if there's 8 and 2? So once you really, below 6 and 4, you get really hard. So you have this problem of you're introducing heterozygotes because of the lack of coverage. And most of those models rely on having a good run, known run of homozygous sites. So we are planning to go back to those samples. So you don't need many samples to do those analyses. You can use 1 or 2 individuals per population, which we hope to do with a PhD student of mine that will give us an understanding of whether that genetic diversity is, that heterozygosity has evolved recently. So like Tasmania, has it been a long collapse, or has it been a collapse in the last 200 years, or was it thousands of years ago? And the genomic data can show you that. The other is to try to estimate something called genetic load, and that's the amount of fitness effects caused by that loss of heterozygosity or novel alleles. So if a population declines slowly over time, then selection will purge deleterious alleles from the population as they go. But if they collapse quickly, then selection can't keep pace with genetic drift. And so we think the rodents of Australia, which had this large population size, large heterozygosity in the 19th century, and then collapsed to very low numbers in some places, might be a signature where we would have increased deleterious alleles. Some other species in Australia, like Tasmanian devils and thylacines, appear to have collapsed to low numbers over a long period of time, based on genomic analyses in them over thousands of years, and so they may have purged those deleterious alleles. So there are some methods that have been pioneered in Europe for estimating genetic load directly from sequences, but they're a little challenging. They involve knowing the conserved state of an allele in other taxa in a comparative framework, and then looking for derived alleles that you assume if they were conserved, their derived state is deleterious, but maybe not. So we're also trying to figure out if there's ways we can infer fitness from the cells themselves, because now we have live cells of these animals, and if we go to 2 different populations, there are methods for looking at the fitness in the cells, whether that reflects the whole organism or not. So yeah, that's the challenge that we're trying to work towards, that you need to infer not all inbreeding leads to the same depression, leads to the same deleterious effects.
I have a stupid question, but when you talked about the king toad,
yes,
you mentioned the species, a northern something.
The northern quoll.
What is the northern quoll? I assume nobody knows. So
No one knows what a quoll is? Okay. So quolls are medium-sized carnivorous marsupials. They're very similar to, like in size to like a marten and they're mesocarnivores of Australian marsupials. The spot-tailed quoll is the largest marsupial carnivore on mainland Australia now, but that lives in the southeast. And yeah, so quolls are something called Australian kids. Sorry?
I just think that the Czech name is Kunovec.
Kunovec is the Czech name for quoll? They have a Czech name?
Marten is Kuna, and
Yeah.
Kunovec is marsupial.
Saying something like Martin-like.
Yeah, Kunovec, yeah, there you go. Yes. Yeah, quolls wasn't really a specialty, and I kind of threw that in because, yeah, colleagues are doing that work.
Thank you.
Sure.
Yeah, so I have a completely different question. First, thanks a lot for the talk, but I was wondering about the work of the taxonomists in Australia. I know that Australia is a huge continent. There are very few taxonomists, and now you are telling us that you are going to use, or you are still already using the remote sensing, which is producing thousands of records.
Yeah.
Who's gonna verify or analyze all these records? Because that's an enormous lot of work.
Well, we do that. We do that before we put it online. And yeah, and fortunately in Southeast Australia, there are a few small species that we can't distinguish, but we can distinguish most of them on camera, which is surprising. Yeah, I know. And if taxonomy, well, so yeah, there's a new one that's come out. So feather-tailed gliders are the world's smallest gliders. They were one species in the genus. We've known for quite a few years there should be two, but the paper officially splitting the two is in press now and it's coming out. And they overlap in Victoria, New South Wales. So yeah, we won't necessarily be able to tell them apart from cameras. And there's a really nice paper in the journal Mammalogy based on American records where they analyzed all the mammal species that people detect on cameras and pointed out the proportion that are indistinguishable on cameras. But it's a minority, but yeah, it's still, I'm not saying the cameras will ever replace going out and collecting specimens. Our hands are tied in some ways. I mean, we are, Australian mentality is not that you should ever, inconvenience an animal. So, yeah.
I want to ask about the timing of the reconstructed colonizations of Sulawesi, Asia, Sahel Shelf, and so on. If the colonizations took place at some period in the past, or under some special circumstances, let's say, or if they take place with the same probability over time.
Well, it's extremely hard to know with the number of colonizations we have, but yeah, the most recent colonizations are of those among those regions about 1 million years ago and spanning back to somewhere 6 to 8 million years ago. And there seems to be no kind of clear jump. There seems to be a shift in taxonomy, that the most recent colonists are all in the tribe Ratini and then in the division Rattus. But yeah, we don't know. I haven't done I think you would want to do a meta-analysis of all the other species that have made these colonizations to see. But I guess that there probably was some period, but there was thousands of years in length. It was more likely during those periods, but our resolution is to hundreds of thousands, if not millions of years. Yeah, hundreds of thousands of years. So the short answer is we don't know. I would say I don't know.
Yeah, I have another question, maybe a more general and intelligent one. This collapse of rodents, of many rodent species in Australia.
Yeah.
So what do we know about the reasons? You said that some happened very quickly after European colonization. So, I mean, in marsupials, it's always said because, well, more modern mammals came in who outcompeted these species. So what did happen with the rodents? Did more modern rodents come in, and is it habitat change by different land use? What are the main reasons?
Yeah, I mean, the short answer is we don't really know. We don't have a good understanding of what caused their decline, but the usual suspects are presented, which is predation by novel predators, foxes, cats, and most of the species that went extinct were in this critical weight range. There's a big study in Australia between, and so that actually larger rodents in groups in a phylogenetically controlled sense were more likely to go extinct than smaller rodents within their group. So in the hopping mice, we lost half the species in that genus, and it was all the larger ones that maybe because they got one suggestion of the predation killed them off. But many of them collapsed much faster than the expansion of predators. And a lot of them collapsed where we now graze sheep and cattle, and where we attempted to graze sheep and it was unsuccessful. So it's never really been quantified well, but that's always been my hunch, that when early settlers were trying to make a living in the harsh environments of Australia and failing, they were eating these chenopod desert environments where a lot of these species lived, and they weren't surviving. So there's also, novel fire regimes, and people have even evoked diseases. But the short answer is we don't really know what their collapse was because we don't have this records to tell us. But some of them were really abundant. They were reported by the first settlers and scientists as like camp pests, and then completely collapsed to either surviving only on an island or to extinction.
Were there some important diseases imported by, let's say, invasive?
It's pure
mammals?
speculation, all that
You
people
don't know
suggested.
anything about
They just say it happened too fast, so that it couldn't have just been this or that. The thing is, we don't really know how fast it happened, because like we were doing these like sort of expeditionary collections as people were like, oh, what's out there, what's out there? And then once we got that, we were like, we weren't doing Grinnellian monitoring. We weren't saying, okay, I need to get an elevational transect because I need to understand the niche of this species, and I need to understand how its distribution's changing. And then, in America, we were doing geographic sampling because we had a Darwinian understanding of species as populations, and so we needed to get geographic variation to split up species. That wasn't really happening in Australia. Intrepid colonist was picking up one or two specimens and sending them back to Oldfield Thomas, and he was describing what he got, and people weren't going back and sampling them again. I mean, just a few weeks ago, I was in the collection, and I noticed two animals that were labeled as swamp rats and have been put in swamp rats, and they just have really long fur, really shaggy, like 60 millimeter long hairs, and they're pale, and swamp rats have short, dark hair, and they're only two specimens I've ever seen like this, and they were collected in 1847 along the coast of South Australia by the then governor of New Zealand, sailing past, and sent to the British Museum. They were sunk in the 1980s into another species. And then when you look up, okay, I said, where are these specimens? And the locality is given is between two rivers, the Glenelg River and the Murray River, and they're huge. They're very far apart. They're more than 150 kilometers apart, so it's in some coastal range. But even when you look up all the specimens that are available in collections, there are no real specimens of rattus from that area. So it may have, nobody went back and said, oh, what's this weird rat? I should go get some more. It was 1847 and never again.
It might be slightly off topic, but you mentioned it. Do you think the bushfires are actually dangerous, besides that human settlements might also burn?
Sorry?
If you think that the bushfires are actually dangerous for the wildlife, besides the risk of burning the human settlements.
Do I think they're at risk for humans?
No. If they are dangerous, besides the risk for humans, the bushfires.
Oh yeah, well there was just a big meta-analysis of the impact of the bushfires that came out in Nature just a couple of weeks ago that shows the impact on species, a mixed response. On the slight average was that they had much, so you can read that paper, there was a massive impact on wildlife. If your question is about what the state will do if the risk to wildlife conflicts with the risk to humans, that's something I toil with a lot, because one of the things about the Black Summer fires is that they didn't really, exceptional cases in Victoria and Malakuta where the Navy had to come and evacuate people off the coast, they didn't affect too many human lives, but it was the largest bushfire in recorded history, covered a huge area and had a massive impact on wildlife. So there was still investment in it, and I do think bushfires and the propensity and the frequency of bushfires is well documented to be increasing. It's one of the key components of climate change that's affecting places like Australia and California and the Southwest, where we're becoming an increasingly fire-prone world. And we're also, in Australia, we've upended millennia of human fire management practices, and then converted things to other things, made new forests, and stopped burning where indigenous people, first peoples, have been burning. And so those changed fire regimes we're just coming to grips with. So there's an appropriate amount of fire and a fire need, and an inappropriate fire. And actually, when you do the literature, we published a review paper on this as well. There is no clear understanding what the appropriate fire is among the scientific community. I think local First People communities know the appropriate fire for their community, but as a global or a national model, we don't know. So we need more people to study that.
You used also once the term black summer fire. Is it a black summer fire or a black fire? So what does the term actually mean? I mean,
so Australia likes to name its fires black whatever. There was like the Black Friday fire and the Black Saturday fire, and whichever day they started. And this one was so enormous that it got a whole season. It was like black.
It's not a type of fire; it's a specific fire event which took place.
Yeah, because it's scorched earth and black, and it's it should be the bloody summer fire or something. I don't know. Yeah. I don't make these terms up, but yeah.
I have a question concerning this, related to these radiations of murid rodents. Because when you show the phenotypes of these radiated clades of rodents, so I thought that you are showing the African murid, because it's very similar in these large African tribes of murid rodents. I saw in African clades there are also these small arboreal rodents, and these wetland rodents, and then partly amphibian rodents. But when you show these large arboreal rodents, in several clades, though, they have half of the tail white, which is also very typical for chrysetomes, which is large arboreal rodent in Africa, but mesomid rodents. It's not murid rodents. So you think it has some. How you explain this? Is it some adaptation to?
Well, yeah, my explanation is going to be any explanation you've heard, and it's really speculation. This is a really common thing, especially among large arboreal rodents, that they have the distal end of their tail is white, and the proximal end is not as dark. And mammalogists don't really know why. We speculate. I think most people think it's because a predator will be attracted to that white tip and attack that. And the animal will get away. But I think maybe one way to test that, maybe we could look at collections I was thinking about the other day, is that you would expect then white-tailed animals to have more naturally docked tails in the wild, and in collections, if you go to them, then animals that have dark tails, because if that's effective, then you would expect, like a lizard can drop its tail and get away, then you would expect to see more of these docked tails. But it's the question mammalogists, you must have been asked it many times, especially herpetologists like to ask me, why do these all have white tails? And, maybe it's just, neutral evolution. There was no reason to maintain their tail anymore. Who knows? Speculation.
To please mammalogists.
Yeah, to give us something to talk about other than skulls.
Question. So thank you very much for the talk. It's very likable, and I know that the rodents are quite frenetic, but your animation was so you can easily slow down a bit. And
well, I can speak for 2 hours if you want me to.
Yeah, we were afraid that somebody would have an epileptic.
Yeah, I should have said a warning. So.
But okay. I also have a proper question. That wetting the traditional taxonomy by the phylogenetics. So I'm curious what's the percentage or amount of the taxonomical error that you found doing those revisions? I mean, oversplitting, overlumping, or just the wrong taxonomical concept?
A percentage I don't know,
amount,
but at least taxonomy tends to have like cultures and groups around it. At least the areas I've been working in, the taxonomists there were pretty good at identifying the species and getting the species right based on the molecules that we've explored so far. Although we keep finding new species, but maybe at the generic level and at the divisional level, which is what kind of non-Linnaean hierarchy we use in murids, they were wrong. Like those carnivorous rats from Sulawesi were thought to be unrelated. Those woolly rats from Sulawesi, Arapepel nomis, they were put in a completely different division with, if you followed my slides, that orange-tailed rat that was in for about half a second up there, it was related to this Pithecia division, which was these arboreal, tufted-tailed rats with weird teeth, and they were put them. But they're part of a radiation of Rattus relatives on Sulawesi. So I think the higher-level systematics was much more informed by the genetics. And then I think the limitation of really fine-scale populations. I mean, I think Josep's done a lot more of this in Africa, of doing fine series with genetics to work out where those population boundaries, and we did some of that with shrews. For the rats, we've done most of the low-hanging fruit first. These very obvious things are completely different, but we're now starting to have to split up species. And we have sunk some genera, so like I mentioned, the Parioromes, that was in its own genus, and there's another genus, Pteromes, but when we sequenced it, we found that it was sister to its type species. So you may not know, so we have type specimens, that's the specimen on which a species name is based, but when you describe a new genus, you also define a type species, the species that defines that genus and is associated with that genus name. And what we found is that Parioromes was sister to the type species of Pteromes, to the exclusion of all the other Pteromes. So we couldn't justify maintaining the name Parioromes. And in other cases, we've elevated existing genera. There was a wonderful genus named Frateromes that we brought back. It means the brotherly rat. And it was described, it was elevated in 1930, not with much evidence, but it was, and then was sunk back into the genus Bunomes, the hill rats. And then genetic data showed us that Frateromes was no more closely related to Bunomes than it was to Pteromes or Linomes and Aeropeplos. So we resurrected the name Frateromes, giving you the mouthful Frateromes fraterorum, the brotherly rat.
So not much oversplitting that you can typically track down a couple individuals suffering, itching, or...
I think there was a lot of the cleaning up of synonymies in Indo-Australian rats through the 70s and 80s that, dealt with a bunch of that. But yeah, some of the higher relationships weren't there.
To clear the things using the good morphology and
yeah, I think there was some
just
very good...
up that, okay, they did it right.
Yeah.
Thank you.
I have also quite general question. When I see these pictures of how many species are described and how many species are still to be described, so I'm not really sure about this estimate. So because I work on rodents, which are the least known mammals, or even a bit on bats. So there are still a couple of species that should be described, but not many. And some decades ago, I worked on insects, taxonomy of some insects, and we described one species, one new species, and I was quite proud that we described one new species of true bugs. But I think that even for insects, they are not so. Is it really true for tropics? Is biodiversity so much unknown, or it concerns some micro invertebrates in soil, or because when I always see these estimates, I'm hesitating if it's really true.
Yeah, I mean, I didn't make this estimate, and I've taken the time to read the literature when I was concerned about it. But there are lots of ways of estimating, like based on the rate of accumulation of new species, of whether like a rarefaction curve, whether you're reaching in a plateau, and then there's other based on where we've sampled, and then I think a lot of the invertebrate collections, if you've worked with invertebrates, you've worked with invertebrate collection curators and collection managers, they can tell you a long list of species in their drawers that they haven't had time to describe yet because there's no specialist working on that group. And so, short answer, I don't know exactly how those estimates were made, but they seem to be consistent among these different groups.
Yeah, I just wanted to know your opinion, because I don't know if you speak with some taxonomists, so they know they have in their collection some undescribed species, but do they really have 80% of species to be described? I think it's.
I mean, the vast majority of that is invertebrates, deep sea invertebrates, remote invertebrates, and other groups that haven't been. But yeah.
It really depends on the group. One example: tiny Prosobranchian subterranean snails from New Caledonia was 5 species described. Then one colleague from Germany did the proper sampling and described 120, like morphologically, new species. So
yeah, Sulawesi a few years ago there was
really
a paper describing 110 new species of weevils from one collection. Yeah, it's not mammals that are driving that, but still, we've had 245 new mammal species in the last 4 years, and more than 1,300 new species since 2005. So that's what 1,300 was about 25% increase in species. So we knew 75% of the mammals as far as we know now, and there's a few percentages left. I don't think we have 80% of mammals left to describe, but like when I went to Sulawesi, I had no idea. I thought I was going there to collect these known genera, and the AMH have been working there for 3 years collecting, and I assumed like most of the stuff was there. And then here were all these weird things we had no idea existed. And on an island, there's 20 million people living on Sulawesi, and it's probably it's 11,000 square kilometers. I don't know how big the Czech Republic is, but it's not a huge island. It's packed with people, and there's still like these remote forests with strange organisms on it, and the invertebrate diversity there is. I work with a colleague there. He describes earthworm diversity, and every time he goes, he gets a new handful of new earthworm species. And earthworms are just a very small tip of the clade of all the arthropods. So, I don't know. I guess
Go back to insects. There's work to do.
Yeah, you have plenty of work to do, though.
No, in rodents, I think we are reaching plateau, so there will be not so many species to describe.
Yeah, well, that's partly why I picked it. I figured it was tractable that we could get to that completion, and then we could provide guidance for what you do with a completely resolved phylogeny. But you've spent a lot of your career probably describing more species than you imagined, and building phylogenies and realizing how much more work there is to do, even if we're approaching it.
So, last question. So thank you once again.
Yeah.